Hello बच्चों, welcome to Hello Biology. This is the 14th lecture of chapter Principles of Inheritance and Variations. Today we are starting with one of the most important Mendelian disorders, haemophilia. Have you ever wondered why some people continue bleeding for a long time even after a small injury? This happens because their blood is unable to clot properly. We just learned that some people continue bleeding for a long time even after a small cut because their blood is unable to form a proper clot. But before we understand why clot formation does not occur in hemophilia, let's first understand how a blood clot is normally formed and what ingredients are required for clot formation. Just like making a cake requires specific ingredients, blood clotting also needs certain essential components. These include platelets, clotting factors, fibrin, and blood cells. All of these work together in a coordinated manner to stop bleeding and help in wound healing. Blood clotting occurs through a cascade where one clotting factor activates the next, finally forming a fibrin clot that stops bleeding. In hemophilia, the most important missing clotting factors are factor 8, hemophilia A, or factor 9, hemophilia B. Due to the absence of these factors, the clotting cascade is interrupted, resulting in delayed clot formation and prolonged bleeding. Now let's understand hemophilia at the genetic level. Hemophilia is a sex-linked recessive disorder caused by a mutation in a gene present on the X chromosome. This gene is responsible for producing clotting factors 8 or 9, which are essential for normal blood clotting. When a mutation occurs in this gene, the body cannot produce enough factor 8, hemophilia A, or factor 9, hemophilia B. As a result, the clotting cascade gets interrupted, and blood fails to form a stable clot. Since males only have one X chromosome, XY, a single defective gene is enough to cause the disease. Females have two X chromosomes, XX, so even if one X chromosome carries the defective gene, the normal gene on the other X usually compensates, making them carriers rather than affected individuals. Therefore, hemophilia is more common in males, while females generally act as carriers and can pass the mutated gene to their offspring. Now let's understand how haemophilia is inherited. The gene responsible for haemophilia is a recessive gene represented by H, and it is located on the X chromosome. Since it is an X-linked recessive disorder, males are more commonly affected than females. A female has two X chromosomes. For a female to suffer from haemophilia, both of her X chromosomes must carry the defective gene, giving the genotype XH. However, such females are extremely rare because this condition is often lethal, and many affected embryos do not survive until birth. If a female possesses only one defective allele, represented as XX, she appears completely normal because the normal gene on the other X chromosome compensates for the defective one. Such females are called carriers. Although carriers do not usually show symptoms, they can pass the defective gene to their children. This is why haemophilia often skips generations and is commonly observed in male offspring. Let's turn towards hemophilia in male. A male has only one X chromosome and one Y chromosome, XY. The hemophilia gene is present on the X chromosome, while the Y chromosome does not carry any corresponding allele for this gene. Therefore, if a male inherits a defective hemophilia gene, XHY, there is no normal allele on the Y chromosome to mask its effect. As a result, the defective gene expresses itself, and the male develops hemophilia. Let's look at some crosses. Cross 1. Here, the mother is a carrier, XH, and the father is a normal male, XY. The carrier mother produces two types of eggs, X and XH, while the father produces X and Y sperms. When these gametes combine, 4 offspring combinations are possible. As a result, there is a 25% chance each of a normal daughter, carrier daughter, normal son, and haemophilic son. Therefore, the overall probability of having a haemophilic child is 25%, and the affected offspring is always a son, XHY. Cross 2. In this cross, the mother is a normal female, XX, and the father is a hemophilic male, XHY. The normal mother can produce only one type of egg, carrying the normal X chromosome, while the father produces two types of sperms, XH and Y. When the mother's X combines with the father's XH sperm, a carrier daughter is formed, XXH. When the mother's X combines with the father's Y sperm, a normal son is formed, XY. Therefore, 50% of the offspring are carrier daughters and 50% are normal sons. Since the mother always contributes a normal X chromosome, no child suffers from hemophilia in this cross. Cross 3. In this cross, the mother is a carrier female, XXH, and the father is a hemophilic male, XHY. The carrier mother produces two types of eggs, X and XH, while the father produces two types of sperms, XH and Y. When these gametes combine, 4 different offspring combinations are possible. The genotype XXH produces a carrier daughter who appears normal but carries the hemophilia gene. The genotype XY produces a normal son with no hemophilia gene. The genotype XHXH produces a hemophilic daughter, but such females are generally considered non-viable and may not survive. The genotype XHY produces a hemophilic son who shows the disease because he has only one X chromosome. Therefore, this cross results in 25% carrier daughters, 25% normal sons, 25% hemophilic daughters, and 25% hemophilic sons. That's all for today's lecture. I hope you now understand what hemophilia is, why it occurs, and how it is inherited from one generation to the next. Don't forget to like, share, comment, and subscribe to Hello Biology for more biology lectures. Thank you and happy learning.